Hi there, I'm Megan, a graduate student in the Overbaugh Lab at Fred Hutch here in Seattle. And today I'd like to tell you about a new method which we have developed to map antibody epitopes and escape mutations, which we call Phage-DMS. And so today I'm going to first go over some background on phage display technology in general, and then how we construct a Phage-DMS library, how we perform experiments with these libraries, and how we analyze our results. And then I will walk us through some quick examples of results that we have gotten with Phage-DMS libraries using either a monoclonal antibody or patient polyclonal plasma. And so first, I will go over the background. So many people attending this meeting are interested in using deep mutational scanning to dissect the binding interactions between their proteins of interest. And so what I study is antibody antigen binding, which is really just one example of a protein-protein binding interaction. And so in order to study this particular interaction, what we have done is we have taken our antigen of interest, whether that be HIV envelope or the spike protein on SARS-CoV-2, and we display bits and pieces of these antigens on the surface of phage. And so phage display is a common tool used to link proteins or peptides on the surface with their sequence inside the phage. So we specifically use the T7 bacteriophage, and in using this system, the protein sequences are expressed as a fusion to the capsid protein on the surface. And so the libraries that I'm going to talk about today are focused on the expression of short peptides, but I just want to note that the size limit of the T7 bacteriophage is actually 1.2 kb, and so they can express proteins of a much larger size. One advantage of the T7 bacteriophage system is that they grow up to really high titers, and that means that you can perform experiments in a really small volume. Additionally, it's important to note that because the phage are grown up in bacteria, this means that they're going to lack any post-translational modifications. So if you're interested in studying an epitope that relies on glycosylation, those aren't going to be added to the proteins when they're made. So that's just something to note. And so to screen antibody antigen binding interactions, we have combined phage display technology and deep mutational scanning. But I just want to say that we're definitely not the first to do this. Other groups have used this, have used phage libraries and deep mutational scanning to, and using other screening techniques in order to dissect the binding interaction that they're interested in. But we are the first to combine phage and deep mutational scanning to finally map antibody epitopes in this way. And so now I'm going to walk you through how we design and construct a Phage-DMS library. And I'm just going to use the spike protein as an example here, but we've previously made libraries with HIV envelope as well. And so you could use whatever antigen of interest you have here. So I'm showing first the structure of the spike protein, which is colored by the different domains here. And we designed our library against just the ectodomain of the spike protein because we're interested in only the parts that are visible to an antibody. And so first we begin by designing wild type peptides, 31 amino acids long. And these peptides overlap by 30 amino acids and tile across the entire length of the protein. And then additionally, at every position, we design peptides that have every possible single amino acid substitution. And so this variable amino acid is located at the central position on the peptide. And so once we have computationally designed all of these sequences, we send these out to a company to synthesize on a DNA microarray. And these get sent to us as an oligo pool. And then we can clone these sequences into the T7 display vector. And this creates our final Phage-DMS library. So in particular for the spike Phage-DMS library that I have built, there were 24,820 variants in total. And so once we build a Phage-DMS library, there are a couple quality control checks that we do in order to ensure that the final library contains a good representation of all the sequences that we originally designed. And so one check that we make is to Sanger sequence a number of individual phage plaques from the library. And this gives us an idea of the percent of sequences which are intact and correct versus those that have indels or other kinds of mutations. And this is because DNA synthesis is an inherently error-prone process, and we find that about 50% of our sequences end up being correct. And this is about on par with other phage libraries that are built from oligo pools. But I just want to note that recently, in like super recent papers, I've seen some phage libraries that report a much higher percent of correct sequences. And this is potentially because, you know, we order them from different companies. So I just want to, you know, take this into consideration if you're thinking about ordering an oligo library, you know, maybe look at some other phage libraries from those companies, and you know, it might be better to aim for one that has higher fidelity here. So another check that we do is to check the uniformity of the library. And we do this by deeply sequencing the final Phage-DMS library. And so something that we have found that actually improves the uniformity of our library is to amplify that original oligo pool by using subcycling PCR rather than just regular PCR. And so subcycling PCR is just a technique where you swap between high and low temperatures during the annealing steps in order to, so that you don't bias against sequences with high or low GC content. And we find that when we use subcycling PCR, we see a lot more uniform representation of the cloned sequences versus using regular PCR, you know, they're more spread out. And additionally, we also saw fewer sequences that were sequenced 0 times. So this means that there are more sequences that end up in the final library. So once we have done these quality control checks of our Phage-DMS libraries, we're ready to run an experiment. And so in order to run an experiment, what we first do is incubate the library with your antibody of interest. And you can use a monoclonal antibody, you can use serum, plasma, whatever you have to work with. And so you allow the antibody to bind to the phage, and the antibody will then bind to the phage that are displaying its specific epitope. And then we add magnetic beads to the mix. And the magnetic beads have a protein on the surface that specifically binds to the constant region on the antibody. And so then we can wash off all of the unbound phage and only then lyse and sequence the enriched phage. And by deep sequencing, we can get a clear idea of the sites of escape. And I just want to emphasize that this is after a single round of selection. And so historically with phage libraries, you know, many people have used biopanning, which is a technique where you select and then amplify and then select and then amplify, you know, sequentially multiple times in order to bring it up to, you know, the enriched phage to a higher representation. But we can get a really clear idea of the sites of escape after just a single round of selection here. So another thing I want to emphasize is that many people using deep mutational scanning will find their method enriches for their variant of interest. However, using this method, what we find is that the variants of interest or the escape mutations are washed off and they're depleted rather than enriched. And so what this results in is we had to figure out a slightly different method of analyzing the data that comes out of these experiments. And so I'm going to now walk you through a toy example just to describe how we analyze the data that comes out of our experiments and how that slightly differs from what other people do with deep mutational scanning experiments. So we begin by sequencing the original input library in order to determine the starting frequencies of any peptides in that library. And here I'm showing peptides that span a random site. And here, these are peptides that span the epitope binding site for a particular antibody. And so perhaps after selection, we would see that the peptides that correspond to the epitope binding site are enriched by the antibody, whereas the other sequences aren't. And so if you calculated the differential selection of this variant and this variant, you would actually find that the differential selection value is the same for these two variants. And so this is clearly not telling the whole story, because the antibody is enriching for the epitope here, these peptides. So in order to account for this, what we do is we then scale this differential selection number by the enrichment of the wild type peptide, the fold enrichment here. And so we just multiply it by the fold enrichment value. And what this does is it emphasizes any differential selection that happens to be taking place at the antibody binding site. So yeah, this is, and this scaled differential selection value is what we report, which is different than just differential selection. And so to summarize, what you get out of a Phage-DMS experiment is two things. First, you can determine the epitope region by just examining the fold enrichment of wild type peptides. So where the wild type peptides are enriched, that's the epitope region. And then you can, within that region, examine the scaled differential selection values in order to find escape mutations. And what we do is we plot the scaled differential selection values in a heat map. And you can see here that red indicates sites where there's reduced binding, and blue indicates sites where there's increased binding relative to wild type. And in these plots, the black dots indicate the wild type sequence at that position. Okay, so this is what the kinds of data that you get out of a Phage-DMS experiment. And so now I'm going to walk us through some examples with some SARS antibodies from either a monoclonal or polyclonal plasma. Okay, so first I'm going to describe the results that we got using a monoclonal antibody called B6. And this antibody we got from the Wiesler and McGuire labs. And we saw their preprint, which showed that this antibody binds to a conserved stem helix domain at the base of the spike protein. And this antibody not only binds to the MERS spike protein, but also binds to SARS-CoV-2 spike protein. And so because this happened to bind to a linear epitope that was defined using this structure, we asked if we could then map the escape mutations for this antibody. And so what we first did was examine just the enrichment of wild type peptides here. So here I'm showing the entire length of the spike protein, and you can see that clearly the antibodies enriching for peptides within this region just upstream of the HR2 domain. And this is in fact the same region that they originally found using cryo-EM. And so if we zoom in to this epitope region, we can then determine escape mutations in that region. And so what you can see here is the red mutations indicate those which decrease binding relative to wild type. And so we can see that there are 5 sites where mutations seem to disrupt binding. And actually this perfectly mirrors the results that they had gotten using cryo-EM. They found that these 5 sites were critical for binding. And one other interesting thing that we noted is that at least at this position, position 1155, there seems to be some mutational flexibility with the antibody, meaning that it tolerates some mutations. The wild type amino acid at this position is a Y in SARS-CoV-2, but in MERS, the wild type amino acid is actually an F. And so you can actually see that it's faintly blue, meaning that the antibody actually binds a little bit better to the peptide with an F at that position rather than a Y. And this also sort of explains why this antibody is particularly cross-reactive, because it seems to be able to tolerate mutations to this position. And mutations at this position are what differentiate many of the SARS, MERS, and SARS-CoV-2 strains. Next, I'm going to describe some results that we have using some COVID patient plasma. And so we partnered with Helen Chu at the University of Washington, and we took some samples from her Harvey study. And so patients were enrolled in this study if they had confirmed COVID infection. And we were able to get samples taken at day 30 and day 60 post-symptom onset. And so first we examined just the enrichment of wild type peptides. So what I'm highlighting in gray here are some regions that were targeted by the antibodies in these people. And what we clearly saw is that the immunodominant regions were near the fusion peptide here and just upstream of the HR2 domain. And this is actually the same region that was targeted by that monoclonal antibody as well. And we saw rarer epitopes being enriched in other places, but most patients were binding to these two regions. And you'll notice that we actually don't see enrichment of epitopes within the receptor binding domain. And this just serves to illustrate one of the drawbacks of Phage-DMS, which is that because you're only displaying short peptides that are 31 amino acids long, you're not able to capture antibodies that perhaps have a complex conformational epitope that relies on distant amino acids coming together and binding. And so, we see in general that our RBD tends to have conformational epitopes. And so we don't really see strong binding there. So I'm just going to zoom in to the fusion peptide region here, and we're going to examine the escape mutations for a few different patients. So I'm just going to start with one patient. This was patient number 1 here. And on the top here is, again, the enrichment of wild type peptides in this region. And these are colored by the different days post-symptom onset. And up here, you can see at 30 and 60 days post-symptom onset, the escape profiles are very similar. So there doesn't seem to be much evolution happening, at least on the antibody side, between day 30 and day 60 post-symptom onset, that the escape mutations are pretty much the same. However, in other patients which targeted this same region, what we saw is that the escape mutations greatly differed between different people. And some of them, the escape mutations were just within the fusion peptide itself, whereas others had, some escape mutations were taking place upstream of the fusion peptide. So if you're interested in examining more of these escape mutation maps, you can either look at our paper, or in our paper, we link to the entire data set, which is now hosted using this really cool web tool that I want to highlight called DMS View. And this was developed by Sarah Hilton in the Bloom lab and John Huddleston in the Bedford lab. And so you can go upload your data onto this site and have it be publicly explorable. And so I've uploaded the data for all of the patients and all of the different time points. And you can select a certain patient and time point and highlight the sites of interest that, and this is just showing wild type enrichment here. And so you select these sites of interest and they are highlighted on the structure over here. And so you can move the structure around and examine where those sites are. And then underneath, they have the scale differential selection values converted into a logo plot format, which is just the same data that's shown in the heat map, just in logo plot form. And so I just think this is a really cool tool, and want to share this with anybody who's interested in having a system for uploading their DMS data and have it be publicly explorable. So that is a cool tool. So what can Phage-DMS do in general? We think it's really cool because we think it could potentially inform vaccine design, because if you understand the specific amino acids that are responsible for antibody antigen binding, you can then hopefully make better, smarter vaccines using that information. Additionally, this can hopefully improve surveillance efforts. As we know, there's many different mutations cropping up across the spike protein, but which ones are important, which ones are ones that we should be keeping an eye on, it's difficult to know, but efforts like ours can help differentiate between, potentially mutations that could be antibody escape mutations. So it can help us keep track of all of these mutations that are occurring across the spike protein. And additionally, it can help accelerate antibody characterization. And what we mean by that is the pace at which people are isolating new antibodies is just really skyrocketed. Nowadays, many people are pulling out tons and tons of antibodies all the time, but the process of epitope mapping those antibodies is a lot slower. And so we hope that tools like this, which can epitope map things really quickly, can help accelerate this and help keep up with the pace of antibody isolation that's happening now. And so finally, I just want to go over the total list of pros and cons for Phage-DMS, just in case you're considering it for your purposes. So one awesome pro is that the system is really fast and really easy to perform. So like I said, it's a single round of selection. From start to finish, an entire experiment takes about 1 week, which is great. It's a super high throughput method. And by that, we mean because we can do this in a 96-well plate format, you can run hundreds of samples, hundreds of antibodies, all in parallel at the same time. And so this has really helped. We've been screening tons of patient plasma samples, and we're only able to do this because the system is so high throughput. Additionally, I didn't have time to mention this, but one cool thing is that you can assay these variants in the context of multiple viral strains all at the same time. So with HIV envelope, there are many strains of HIV. And so how do you choose which strain to do your deep mutational scan against? Well, you don't really have to. You can choose many strains. And so we actually had 3 different strains in our HIV envelope libraries. And additionally, I mentioned this before, but we only need a tiny amount of antibody sample in order to run these experiments, which is great when you're working with like really limited sample. So if we're talking about the cons of Phage-DMS, one big one is that so far it can only map linear epitopes that are expressed on, in our case, 31 amino acids. Additionally, they don't have any post-translational modifications. Right now, this is the length of the peptides is limited by DNA synthesis technology, but as this improves, as our ability to make longer and longer DNA molecules improves, that we can increase the size, the length of these peptides expressed on the Phage-DMS libraries. And additionally, one drawback is that high error rate during the DNA synthesis step, which means that about 50% of your library is correct. And that means when you do these sequencing runs, about 50% of your reads are going to have to be thrown out at the end. So it's sort of wasteful. But overall, we think it's a really cool system, and we'd love to see more people use it. So if you're interested in learning more about Phage-DMS, you can go see my papers on this. So we've published the method in this original paper. And if you're interested in learning more about those COVID patient plasma samples, that is in this bioRxiv paper, which is currently under review. And so finally, I want to acknowledge everyone in the Overbaugh lab who's been super helpful for getting Phage-DMS up and running. Additionally, the Matson group has been absolutely essential in, as these experiments have gotten bigger and bigger and the computational load has increased, the Matson group has been really great at coming up with a system for us to manage and deal with all this data. Additionally, we'd like to thank the Chu lab, which has given us a ton of samples to work with, the Bloom lab, who's our deep mutational scanning gurus, the Bedford lab for developing that really cool DMS view tool, and the McGuire and Wiesler lab for giving us the B6 antibody. So with that, I will say thank you and hopefully be able to take your questions.